Okay, so good evening, everybody. So for those of you who don't know me, I'm Ashley Dias from the Ministry of Environment. So actually our name has changed. So we're now the Ministry of Agriculture, Climate Change and Environment. And I will be the chair for this evening session. So just to start off, two main rules for today's presentations. So each presentation will go on for about 10 to 15 minutes each, and then we will have a 4-minute Q&A at the end of each presentation. And before we start, I will be introducing the presenters, and so we will move on from there. So we have three presenters for this evening. And the first to present is Noam Vogt-Vincent. So Noam is a second-year PhD student at the Department of Earth Sciences, Oxford University, investigating marine dispersal around ocean islands in the Western Indian Ocean and Japan, and will later be focusing on Aldabra Atoll. He previously studied geology as an undergraduate at Oxford and has carried out research on coral habitability in Japan during the last ice age and ancient ocean currents in the North Atlantic. So his presentation will be about mapping coral population connectivity and ocean currents to inform management and policy of the coral reef systems in Seychelles. So over to you, Noam.
Thank you very much. All right, is my screen visible? I'm assuming it's going to be visible unless anybody.
Yes, it's working.
Thank you. Cool. Thank you very much. All right. So yeah. So I'm Noam, and I want to talk about the work that I've been doing recently, looking at modelling the connectivity of coral reefs in Seychelles and the wider southwestern Indian Ocean. To start off quickly with some quick theory. So corals, once they're established on the ocean floor, are obviously not particularly motile organisms. So they don't move about very much. But coral larvae are motile because they can be transported for very, very large distances by ocean currents. And that's very important because if a coral larva travels from the parent reef it came from, and it travels a long distance to a different reef and ends up settling in a different reef, connectivity is established between those two reefs. And if you have a consistent flux of larvae traveling from a source reef to a sink reef, then you've got connectivity between those two sites. And this connectivity has lots of important ecological implications, which is really important for marine spatial planning. So for marine spatial planning, it's important to have an understanding of the connectivity of reefs. But measuring this connectivity is very difficult because coral larvae are really tiny and they travel for extremely long distances. So measuring connectivity directly is generally infeasible. So you usually use either genetic methods or simulations, computer simulations, to predict this connectivity. And these two methods, genetic methods and simulations, are complementary. And April Burt, who most of you will know, has been using genetic methods to map out connectivity of reefs in the Seychelles. And I'm focusing instead on simulations. And after our projects are done, we're going to compare our two connectivity results and see how they differ and how they're similar. But in this talk, I'm going to be focusing on simulations. So I'm not going to go into detail about how you actually go about simulating coral larval dispersal through simulations. But basically, you have to decide where your larvae are coming from. So I should say, first of all, so obviously, rather than using real coral larvae, you're using virtual coral larvae. So we're representing our coral larvae as kind of virtual larvae in a computer program. And you decide where your larvae are coming from, and then some basic behavioral parameters. So things like how long does a coral larva have to be in the water column until it's competent to settle in a different reef, or how long can your coral larva actually survive in the water column. And then you combine all of this information with ocean current data, and the computer predicts where the ocean currents transport your coral larvae. And then you repeat this for millions and millions and potentially billions of virtual larvae from all of your different reef sites in your region of interest. And then once the computer has predicted where your coral larvae are going, it then calculates the likelihood of a coral larva going from one reef to another reef. And that directly gives you connectivity information. So we'd often present this in a connectivity matrix like this. And this matrix is really useful because, for example, if we wanted to know the chance of a larva going from reef 20 to reef 5, we go to 20 on the x-axis and then go up to 5 on the y-axis, and that box is green. And the green means there's about a 1 in 1,000 chance of the larva going from site 20 to site 5. And 1 in 1,000 might sound like a very, very low chance, but actually, if you bear in mind that literally millions of larvae are being released from every single, or being generated from every single spawning event, a 1 in 1,000 chance is actually, that represents a very significant flux of larvae from one site to another site. So this kind of connectivity matrix is what we want to end up with. So step number 4, ocean current data, is actually the most difficult step here, arguably, because trying to predict what the ocean currents are doing in a large region is very challenging. And there are 2 ways of obtaining this data, usually. Satellites is one option, or computer models is another option. And in short, satellites it's relatively easy to get ocean current data from them, but it's very low resolution. So if you look at the top image here, it's very pixelated. And this image shows surface ocean velocity, and it's very pixelated, and that's because of the low resolution of ocean current data from satellites. Whereas the bottom image shows the ocean currents predicted by a model, so a simulation that I'm running. And as you can see, it's much higher resolution. So that's the major advantage of a computer simulation, that you can get a really, really, really high resolution. The disadvantage is that these are not observations, these are predictions by a computer. So you have to then compare this data to observations to make sure that it's sensible. So the aim of the project is simulating coral larval dispersal within this region here, highlighted or bounded by the white box. So everything from the East African coast over to the British Indian Ocean Territory in the east, and then centered in Seychelles in the middle. I'm simulating ocean currents at a 2-kilometer resolution for 25 years. And this is the largest and highest resolution simulation that's been carried out for this region. And then once the simulations are finished, I'll be calculating connectivity matrices. And as I mentioned before, then comparing these connectivity matrices with April's genetic data and seeing at the similarities and differences between the modeled data, the modeled connectivity predictions, and the genetic connectivity predictions. So I've talked a lot about theory here. So I want to show you some actual simulations. So here I am releasing several 100,000 virtual coral larvae from Aldabra Atoll in the southern Seychelles. And I'm letting these larvae drift around in the ocean for 2 months. And the colors, so the background shows bathymetry, and the animated colors show the density, so the concentration of virtual coral larvae as we go forward in time. And as you can see, the larvae travel westwards predominantly from Aldabra, because the prevailing ocean currents around Aldabra are to the west. And within about 2 weeks, they have reached the East African coast. And after about a month, we've already, coral larvae coming from Aldabra have already managed to reach many of the coral reefs on the East African coast. So we would predict that Aldabra is a really important source of coral larvae for reefs in the East African coast, and also in the Mozambique Channel. And here we're looking more broadly at several different sites in this kind of southern Seychelles region, and all of these islands are kind of in the path of the powerful westward ocean currents. So these are the southern equatorial current and the northern Madagascar current. And as a result, all of these islands, the coral larvae are rapidly swept westwards. And within a few weeks to about a month, they have managed to reach many reef sites along the East African coast. So all of these, so this predicts that all of these reefs in the southern Seychelles are probably going to be really important for downstream connectivity for islands that are downstream. So here we're now looking at Mahe, and I'm releasing coral larvae from Mahe, and they're being transported predominantly eastward because the predominant ocean currents, at least at this point in the year, in the granitic Seychelles are eastward ocean currents, but they're not getting that far away from Mahe. So compared to the reefs in the southern Seychelles, they're not traveling particularly far away. So we would predict as a result from this that the reefs in the granitic Seychelles are probably more genetically isolated than the more southerly reefs. And here we're looking at so the granitic Seychelles and also the Amaranth and Alphonse. And as you can see, all of these reefs, they don't travel to any other major island group. So this would predict that at least at this point in the year, these reefs are more isolated than the reefs in the south. So these simulations, these are just examples from one single release in one single point in time, but ocean currents can vary over time. So rather than just doing this for a few selected coral reefs for one selected 2-month period of time, I'm going to eventually repeat this for all of the reefs in my entire domain, and then for 25 years. And then we'll be able to assess if these connectivity patterns are consistent with time. So for example, if we get back here, if my computer doesn't freeze, is this pattern of connectivity from Aldabra, is that just happening once here in this one snapshot, or is this consistent over a longer period of time? So finally, all of this data will be made publicly available. The kind of main thing I'm applying this to at the moment is coral reef connectivity, but this can also be applied to other applications. So we're also interested, for example, in applying this to to the dispersal of marine debris. So for example, trying to work out where debris arriving at Aldabra actually comes from. So there was a great talk the other day about the Aldabra cleanup project, and using these ocean current simulations, we can try to predict where that debris is coming from. So there are all sorts of applications for, that you can apply this ocean current data from. And if you'd be potentially interested in using this, I'd be very interested in hearing from you. So that is me done for the time being. And here's my contact information. I'm very happy to hear any questions.
Okay, so thank you, Noam, for this very interesting presentation. So if any attendees have any questions, please drop them down in the Q&A section. So just to get things going, Noah, you've mentioned that your results can assist us in working on our management and policy decisions. So actually currently at the ministry, we have appointed some consultants to work on a coral reef policy for us. And so how, on your side, how would you feel that your data can assist us maybe? Because one of the objectives of their work was to define measures to reduce the threats to coral reefs, as well as prioritize actions for monitoring and reporting. So what can you advise us maybe on how we can proceed? Because we're quite at an advanced stage, and maybe if you have any guidance on how we can improve our policy maybe.
So I'd be very happy to discuss that with you guys in more detail at a later point. And this work that I've got here is all preliminary data, so I haven't got any conclusive data from this yet. But as I kind of briefly said at the start, what this information tells you is the likelihood of larvae going from one reef to another reef. And that data is important for conservation efforts because, for example, if you know that a particular reef site delivers a large supply of larvae to many other different reefs, then that reef could potentially be acting as a really important lifeline for other reefs in that region. So that might potentially be a priority for conservation efforts, because if that reef suffers a really bad mass mortality event, for example, that lifeline is gone for many other downstream reefs. Whereas if you have a reef which is getting larvae from lots and lots of other different reefs, it might be more resilient to a mass mortality event because it's able to obtain larvae from many other reef sites. And even if some upstream reef sites undergo a significant mass mortality event, it's still receiving larvae from other reef sites. So basically, this information can help guide policymakers to predict which reefs might be a priority for conservation efforts and which might be more resilient to environmental change.
Okay, thank you. So you've received some questions.
Yes.
I will start with a question from Denise. She's asking, if I understand correctly, April's genetic connectivity work will be able to provide ground truthing for this method, for these models in Seychelles. Are you looking at obtaining ground truthing from other regions, such as coast of East Africa?
Thank you for the question, Denise. So firstly, comparing this data to genetic data is partially ground truthing, but also the genetic data contains some extra biological effects which the computer simulations don't tell you about. So it also tells you, looking at the differences between simulated predictions, which are mainly based on ocean physics, and the kind of ultimate genetic predictions, also tells you some interesting things about the biology of coral larvae. But yeah, primarily it is there for ground truthing, and ground truthing of data from other regions like East Africa or Chagos would be very interesting. I'm some way off from being at that point yet, but ground truthing it with a wider region would obviously be great. So yes, that would be good if I can do it.
Thanks. Another question is from April. She's asking, could you use this for lobster or fish larvae dispersal?
Thank you, April. So the answer to that is possibly. The complication is that lobster and fish larvae swim a lot more strongly than coral larvae. So coral larvae to first order go where the ocean currents tell them to go. Fish larvae and lobster larvae have a bit more autonomy, which can actually be really important. So it isn't very possible to do this for fish larvae and lobster larvae, but the biological aspect is a lot more complicated. There are potentially more uncertainties, and that is not something I've looked into a great deal already, but potentially it is possible, yeah.
Okay, thank you. Next question is from Jean-Pierre. So he says, your talk is very interesting. And do you know if there's any current work to improve satellite data resolution to help with real life correlation to the simulations?
So this is a really good question. The issue is that, so if I go back to the satellite image, this low resolution isn't from the satellite. It's partially from the satellite, but more importantly, because of the way that the satellite calculates these velocities from what it's measuring, there is a fundamental physical limit on the resolution of data that it can obtain. So regardless of the resolution of the data of the satellite, you cannot obtain ocean current data from the satellite that is directly, at least, that is a much higher resolution than the image at the top. There are certainly other things that you can do to improve your ability to use the satellite data. So first of all, on large scales, it may be true that even this very coarse satellite data is adequate for predicting connectivity. We don't know this properly yet, and actually that's one thing I'd be interested in testing in this study. And there are potentially other correlative studies that you could do. But in terms of improving the resolution of the satellite, you can't do that here because of the physics involved in calculating it from the satellite product.
Okay, all right, great. So we have time for two more questions. So one question is about, can this model be used to predict the appearance of rare genetic diversity?
Right, so I am not a biologist, and I probably cannot answer this question particularly well because I don't particularly know what rare genetic diversity means. However, what these models can do is that they can predict, because we are simulating huge numbers of coral larvae, we can predict the likelihood of long distance dispersal events, which might be very, very low probability, but which are still a large enough probability such that they are over kind of evolutionary timescales important. So, for example, one question that we're interested in is if my skin doesn't freeze, which I think it has. One question we're interested in is there any significant connectivity between Chagos, the Chagos archipelago, and Seychelles? So there's probably no particularly strong connectivity between the two. But even if there is, oh, here we go. Even if there is a tiny, even if a tiny fraction of larvae manage to get from Chagos to Seychelles or vice versa, then for establishing genetic connectivity, that could be really important.
Noam, and our last question for you is, will you parameterize your model with larval behavior for a variety of different coral species?
Yes, so the answer to that is yes. So the two species I'm particularly interested in are the two species that April is looking at, and we do have data on the mortality, for example, of those two species. So we definitely will be using that. In terms of other biological parameterizations, things like I've forgotten the word for that, but behavior, for example, coral larvae can swim vertically a bit, and that could potentially be important, and we may take that into account. I'm not sure yet, but probably the most important parameterization for coral connectivity is mortality, so the lifespan of a larva in the open ocean, and that we definitely will be using. And to very quickly answer the question from Timothy, we're using computer simulation. So we're not using I'm not using satellites at all for this. So indirectly for ground truthing, yes, but for the actual simulation, for the actual dispersal modeling, I'm purely using computer simulations, not satellites.
All right, so thank you very much, Noam, and for everybody who asked questions.